As senolytic drugs transition from experimental models into human trials, one of the most urgent questions is whether they can be used safely in older adults, especially those with multiple chronic conditions who may benefit the most. The elderly population often lives with a complex burden of comorbidities, polypharmacy, and reduced physiological reserve, which can amplify side effects or drug interactions. Yet these are the same individuals for whom senolytics hold the greatest promise, a means of targeting the root cellular processes driving frailty, chronic inflammation, osteoarthritis, cardiovascular dysfunction, and even cognitive decline. So far, the early evidence is cautiously optimistic, suggesting that when used intermittently and carefully, senolytics may indeed be safe and well tolerated, even in vulnerable populations. Senolytics are a unique class of drugs designed to selectively eliminate senescent cells, which accumulate with age and secrete inflammatory molecules, matrix-degrading enzymes, and other harmful signals collectively known as the senescence-associated secretory phenotype, SASP. These cells are implicated in a wide range of age-related diseases, and their removal in animal models has shown improvements in lifespan, physical function, and resilience to stress. But translating that success to humans, particularly older ones, requires a careful balance, clearing harmful cells without disrupting tissue stability or triggering systemic inflammation. Initial human trials have provided encouraging data. A landmark pilot study conducted at the Mayo Clinic and published in eBiomedicine, 2019, evaluated a combination of dasatinib, a tyrosine kinase inhibitor, and quercetin, a flavonoid with anti-inflammatory properties, in patients with idiopathic pulmonary fibrosis, a severe age-related lung condition. Participants, mostly older adults, received 3 days of senolytic therapy per week for 3 weeks. The treatment was generally well tolerated, with mild and transient side effects such as nausea and headache. Importantly, some functional improvements in walking distance and mobility were observed, despite the short treatment duration. Another small-scale, open-label study in adults with diabetic kidney disease also showed that senolytics could reduce the burden of senescent cells and pro-inflammatory SASP markers in adipose tissue. Participants reported no major safety concerns, and side effects were again mild. These early-phase results, while preliminary, suggest that short, intermittent dosing schedules, sometimes called hit-and-run therapy, may be key to minimizing risk. Unlike many chronic medications, senolytics do not need to be taken daily. Instead, they aim to reset the tissue microenvironment, allowing a recovery period between doses. Of course, safety concerns remain, particularly with agents like dasatinib, which was originally developed for cancer and can affect bone marrow, platelet counts, and immune function if used at high doses or for extended periods. But in senolytic protocols, the doses are typically much lower and less frequent than in oncology. In preclinical models, even a single round of senolytics has produced lasting health benefits, suggesting that repeated exposure may not be necessary and therefore risk may be more manageable. Still, older adults are a highly heterogeneous group. Some are robust, physically active, and biologically younger than their age. Others are frail, on multiple medications, and have impaired organ function. This variability underscores the need for personalized senolytic strategies. For instance, a frail 85-year-old with renal insufficiency may require lower dosing, more extensive monitoring, or a gentler senolytic agent such as fisetin, a naturally occurring flavonoid with senolytic properties that appears to have a favorable safety profile in both animal studies and early human trials. Several clinical trials are currently underway to test senolytic safety and efficacy in broader aging populations. One of the most notable is the AFFIRM trial, examining whether fisetin can reduce inflammation and preserve function in older adults during hospitalization. Another is targeting Alzheimer's disease, with senolytic therapy aimed at clearing senescent glial cells that may be driving neuroinflammation. These studies include participants over 70, many with comorbidities, and their results will provide critical insight into both tolerability and outcomes in real-world aging. There is also growing interest in using biomarkers to guide senolytic treatment, such as P16-INK4A expression, circulating inflammatory cytokines, or SS-galactosidase activity. These markers may help identify individuals with a high senescence burden who are most likely to benefit, and perhaps flag those at greater risk for side effects. In the future, routine blood tests could indicate whether a person's tissues are being overwhelmed by senescent cells and whether it's time for a clean-out cycle with Senolytics. Perhaps one of the most reassuring aspects of senolytic development is that it isn't attempting to hijack metabolic pathways or suppress symptoms. Rather, it's a root cause intervention designed to remove cells that should have already been cleared by the immune system. When dosed correctly, senolytics appear to enhance tissue function without impairing healthy cells, and their intermittent use minimizes cumulative toxicity. In sum, while much more data is needed before senolytics become mainstream treatments for older adults, the early safety profile is promising, especially when matched with thoughtful dosing, monitoring, and patient selection. The approach doesn't promise immortality, but it may help restore a measure of vitality to aging tissues by clearing out cellular debris that accumulates with time. For elderly patients who currently face a long list of medications that manage symptoms but don't reverse the process, senolytics could represent a new kind of therapy, one that targets aging itself safely and precisely.